So there's many thanks this morning. One, thanks Matt for organizing a great meeting. Thanks to Lark, who's out, I guess, in the entryway for the entertainment last night. I thought that was the best part of the meeting, to tell you the truth. I'll just do some further thanks along. I mean, the other thing I should thank is the funding agency, which is actually a huge thanks these days, given the tight funding situation. But a young lad asked me not too long ago why I study dioxin. And really it started probably right in the mid-1970s. I was living in St. Catharines, which is downstream of the Niagara River, and we got a lot of buffalo news. And in the news right around then was Love Canal, and it sort of piqued my interest a little bit. And I was also one of those kids, when I was in, I was 10, 11, 12, I used to watch Meet the Press, okay, so that gives you a flavor. So I was also primed for that because of Nixon's war on cancer. And so it was right around then that I really started getting interested in research, to tell you the truth, believe that or not. So if you fast forward that now, 5 or 6 years, I go to Guelph, the University of Guelph, which is just outside of Toronto. And this is also an opportunity for you guys to caffeine up, because we're really going to get into some heavy stuff in a few minutes. But anyway, and lo and behold, Steve Safe is there. And as an undergraduate, I'm walking around the halls and I see all these posters about PCBs and dioxins, and I'm going, hey, Love Canal, that's interesting. So for years I tried to get into his lab to do research, and it wasn't possible because by that point in time he was down in Texas. But there was still, the opportunity every once in a while he would come up and visit his people. So one of those times I snared him in his office and I said, hey, can I do graduate work with you? And he said, yes, but you got to go to Texas. 6 months later, I'm baking in the sun down there, sweating like a pig because, I'm Canadian and we're not acclimatized to, 100-degree temperature swings. So that was like the 80s. 20 years later, I'm at MSU, and my chair walks into my office 2 years exactly to the day after being hired and saying, your 2 years of non-teaching are up. This year you're going to be teaching metabolism. I'm going, are you kidding me? Metabolism? I'm a toxicologist. I haven't looked at metabolism since I was an undergraduate. He said, don't worry, it'll be good for you. 10 years later, almost 10, 15 years later, I'm up here today giving a talk on essentially genomics. And I want to thank Bill Smith, my chair, because he was right. And what I'm going to talk about today is really the integration of various genomic approaches to further investigate the mechanism of action of TCAD. So we've seen this slide already, and so I want to go through this quickly since I just burned about 5 minutes of my 20 to 25 for this presentation. But essentially, TCAD passively diffuses into the cell, binds to the H receptor in the cytoplasm, causes dissociation of associated proteins. It translocates into the nucleus where it heterodimerizes with the aryl hydrocarbon receptor nuclear translocator. It then has the capability of binding to DNA, and it binds specific response elements. And these are referred to as dioxin response elements, or DREs. This causes changes in transcription. They can go up or they can go down. And obviously changes in protein. And if I was going to change this slide or update this slide, these changes in protein, some of them are going to have enzymatic activity, so there's going to be some metabolite changes as well. But eventually, a culmination of those is going to result in your physiological and cellular responses, and some of those responses are going to lead to the toxicity. So the thing that we want to do, and this is sort of an overview of what we've been doing for the last 10 to 15 years, is really trying to understand what's going on at the gene expression level, in the gene space, in the protein space, and not so much here because this is a little bit too complex for what I've got, my capabilities, and the metabolomic space, because really changes in gene expression are going to cause changes in protein. Some of those proteins are going to come back and cause changes in gene expression. Some of those proteins are going to have enzymatic activity, cause changes in metabolites, which could affect protein as well as affect changes in gene expression. So really, if you don't integrate all of this information, then how can you really have a true appreciation of the comprehensive activities that's ongoing as a result of exposure to this chemical? So what we've done is we've chosen the female C57 black mouse as our preferred model. We've done time courses and we've done dose response, and today I'm going to primarily focus in on a dose response study that we did. It was a 28-day study. It was, the doses are indicated here from 0 to 30 micrograms per kilogram. And we dosed those animals every 4 days. And this was an oral gavage dose. And we achieved steady state levels of the dioxin. At the end of the study, we just harvested a whole bunch of different tissues for different kinds of analyses. Obviously, we collected tissue for gene expression analysis, collected tissue for ChIP, and I'll talk a little bit more about what that is specifically. We did metabolomic analysis, targeted as well as more comprehensive. We collected tissue for histopathology. We did some clinical chemistry on these animals. We looked at tissue weights, obviously, and we measured TCED levels. And I'm not going to talk about everything, given the time that I've got, but I'm going to give you enough information, I hope, to convince you in terms of what the potential mechanism for the toxicity of TCED is in the mouse liver. So the first response that we looked at was really the histopathology. And there was 3 things that really were striking in terms of that. And the first stain was actually the H&E stain. This one doesn't really show it that well, but it was the steatosis, the fatty liver response, the accumulation of fat that was occurring within that. And that was actually demonstrated and further verified by oil red O staining. Now what we've been able to do is actually take that and do digital pathology on that to be able to quantitate the level of oil red O staining. We've been able to go beyond that, not just measure how much fat is accumulated, but also be able to differentiate between the micro as well as the macro, and that's what that slide is on the bottom. You can see how there's a sort of a dose-dependent increase in that. Now it's sort of an artificial measure because as we further investigated that, there's actually just there is no differentiation between a micro and a macro. The size of the vesicles just continue to increase. And what we were looking at as a result of this is we could see that there was this normal distribution of sizes. There wasn't a bimodal distribution where the first half would be micro and the second half would be macro. We further verified the immune cell infiltration with the F480 staining. You see an increase, especially at the high dose. And there was also some undulation on the surface, and Jack Harkema said, well, you might as well do some Picocereus red to look at fibrosis. And so we did that, and right around the time we finished this, a publication, or actually a couple publications came out that verified that there was fibrosis occurring as well. So we've got what I'm going to call the sort of the apical endpoints that we're interested in that we think are contributing to the overall toxicity that's occurring. We then looked at it from the other way around. We said, okay, we know from the mechanism of action that the H receptor has to bind to the dioxin response element in order to cause these various effects. And we know as a result of the availability of the human genome as well as the mouse and the rat that we should be able to computationally be able to search and identify every gene that has a dioxin response element. So the first thing that we did was we identified the 5 base pair core, and then we extended that out to 19 base pairs so we can get a score. We could do that with a position weight matrix. And essentially the results are indicated in this table here. I won't do what Jim did. I won't disenfranchise this half of the room. And so if you look at the mouse sequence here, you can see that the ref-seq, which essentially represent a gene, there's about 24,000 genes that are represented within the mouse genome. Or there's 24,000 ref-seqs, about 20,000 genes that are represented. Approximately 19,000 or almost 20,000 essentially had that specific DRE core. But if you extend that core out to the 19 base pair dioxin response element based upon functionality of known characterized ones, there's only about 13,000 are actually considered functional. So we can use that information now to see whether genes that are responsive actually have what we call a functional DRE core and determine whether there's actually that response or that change in expression is actually due to the H receptor. So the second aspect now is that we can actually go and we can look at where the H receptor binds. And this is where this ChIP assay comes in. ChIP refers to as chromatin immunoprecipitation. And what you can do is you can take the H receptor, activate it, let it bind to DNA, and then capture those sequences that it binds to, sequence them, and determine where in the genome that H receptor is located. So if you do that, and in this study we did that for 2 hours and we did that for 24 hours, we see that there are about 22,000 enriched AHR regions within the mouse genome. At 24 hours, that decreases to about 13,000 enriched regions. But you would think that if a gene is going to be expressed and it's going to be mediated by the AH receptor. The AH receptor would bind close to that gene, and under the peak that it's binding, you would hope to find a dioxin response element. So we can further investigate the mechanism of action that's associated with the effects. Is this a primary effect? Is it a secondary effect as a result of activation of the AH receptor? The third component was really hepatic transcriptomics. And so here we're using RNA sequencing. So we took our dose response, and this is really the 28-day study. We looked at 7 different doses, and then we took 5 animals from each dose, and we did sequencing of these. For each one of those samples, we looked at 30 million reads, and that's probably a little bit excessive. We could probably get away with 25 or even 20. From that, we can throw out about a third of the data, because the reads did not align it to anything, and that's pretty typical of most RNA sequences. So we had about 21 million reads to work with on average per sample, and those 21 million reads could be mapped back to about 18,000 genes. And so that's about 18,000 genes that are expressed within the liver samples across the 0 to 30 microgram per kilogram dose. Of those ones, and so the cutoff here was that we had to have at least 5 reads that could be mapped back to a specific gene. If it was only 4, then we wouldn't consider that to be sufficiently enough reads, and so we would not include that gene. If we look at those 18,000 genes and we apply what we use an empirical Bayes approach to identify differentially expressed genes, because then it gives us a probability as opposed to a hypothesis test. And we can set the standard, the P1T value, to anything that we want, and we're not violating any kind of parametric analysis. So on a parametric analysis, you have to say that, you know, if above the 0.95 level, the gene, you have to accept the hypothesis that it's differentially expressed, and below that, you have to reject it, and we don't have to do that with the Bayes analysis. So it's a statistical trick that the parametric guys sort of cringe about, but the empirical Bayes guys really like it. We came up with about 200 genes that were differentially expressed. Here are some of the examples that we came across, and I'm going to talk about more of these examples as we get further into the analysis. We had evidence of oxidative stress. Boy, and I have to remember some of these acronyms. Glutathione peroxidase, xanthine dehydrogenase, hexose or glucose 6-phosphate dehydrogenase. I'm impressed already. And then I forget this one, but that's another one that's involved in glutathione production. When we get into the lipid metabolism, we got, well, that's a lipase, so it's involved in terms of the triglyceride hydrolysis. This one's involved in phosphatidylcholine production. I forget it, boy. This is another one involved in 1-carbon metabolism, and some of these things we actually talked about before in terms of Dean Jones's talk yesterday, and one that's involved in acetyl-CoA production. In terms of immune cell infiltration, we have gene expression that's associated with activation. And in fibrosis, we have expression of COL1A1, which is obviously what's going to be responsible for laying down collagen. So obviously the next step then, we've looked at the DREs, we've looked at where the H receptor is binding to the DNA. We've seen that there's some changes in expression of these genes. Does that change in expression actually have a functional consequence? In addition to just being expressed, is it actually causing changes in metabolites? And here we did a targeted metabolomic approach. And in this approach, what we essentially did was we looked at 200 metabolites that we identified, and I have standard curves for, so that we have confidence in terms of what we're able to detect. And that gives us a snapshot, or a limited snapshot, in terms of what's changing in terms of metabolites within these 3 different compartments. And so we've actually looked at liver extracts, so that'd be aqueous, and obviously all these are aqueous. We've looked at serum, and we've looked at urine. And of those, you can see that approximately 100 samples or 100 analytes changed in the liver extracts, about 45 within the serum, and another 100 within the urine. And so what we want to do, okay, well, let's look at this. If we look at the liver, some of the extracts that were changing were oxaloacetate. The red is going to indicate that that metabolite increased in terms of its level, whereas green, the acyl-CoAs, decreased. So that's going to give us some indications of what's happening as a result of those changes in expression. In the serum, we saw hypoxanthine and xanthine decreasing dramatically. I mean, this was about a 50-fold decrease. So these are what I refer to in the lab as the low-hanging fruit. Go for the low-hanging fruit first, build your story around those, and then see if you can find the interconnections between them. And then in the urine, we saw ascorbate acid increase in a significant way, and there was a decrease in lactate. So then the question becomes, what does all this mean? So you've got all this data, very impressive, it's all exciting, but it's still all descriptive. And what we refer to this as previously is discogenomics. Disc goes up, disc goes down, and and nobody really cares beyond that, right? Okay. Remember I said, thank you, Bill Smith, for the assigning me metabolism, because what I get from about the end of February to the end of March, I got 6 weeks of 400 undergraduate students that are non-biochemistry majors. And I'm responsible for teaching them carbohydrate metabolism, lipid metabolism, and protein metabolism. And it's perfect, because if I didn't know that, and I'd be, I could test you guys, and I bet you don't know it as well as you would hope to as well, I'd be really in trouble in terms of trying to understand what this. Now, I can't take all the credit because I've got some graduate students in the lab. I'm just the eye candy, right? It's the people in the lab that actually really do the work and come up with the, with these kinds of things. So here's where the heavy lifting comes in. This is what we've got. So in total, what this represents, this network map represents, is 338 genes. Now, all those genes are changing, but they fit into a pathway, an overall pathway that's affected. 98 of those genes were induced, 48 of them were repressed. 105 of them showed that there was a dose response. With gene expression, we can do a computational model to identify the best fit for the curve and come up with an EC50 associated with that, and that will become important later on. With the CHIP data, we know that 148 AHR receptors associated with those genes, so that gives us some confidence that, hey, the H receptor is actually doing something within those specific pathways. And there are 307 DREs. The DREs are becoming less important because now we know, as a result of that CHIP as well, that not all binding of the H receptor to the genomic DNA is mediated through a DRE, that you can have something that's called tethering, or the H receptor will actually piggyback on another transcription factor to bind to that promoter region and cause changes in expression. If we look at the metabolites, these spheres represent 95 metabolites, 20 are increasing, 21 are decreasing, and 33 show a dose response. Now in this case, the dose response is just by eye, because we only looked at 3 doses, and that's because, oh, that's another story and I don't want to get political, not right there, now anyway. In total, this represents 15 different pathways. 10 are upregulated and 5 are downregulated. So the green represents stop in genomics, just to confuse you further. The red means that it's go. So let's look at this, and I know that you can't read this, and it's not intended for you to read it because it's an overall view. But if we take a snapshot of these two things, and actually there's 2 things I want to get into a little bit further today, is one, the NADPH pool and its overall role in terms of contributing to TCD toxicity, or how the cell is responding, and specifically in the mouse liver, and the acyl-CoA pool, and specifically acetyl-CoA. And looking at these 2 pools will then allow us, or allowed me and the people in my lab, to sort of further investigate, well, how are these things interconnected in terms of how is the hepatic cell responding? So if we look at the NADPH pool, what we see is that there's a decrease in the pool itself. But if you look at the pathways, that doesn't necessarily make sense. And what it's really doing is it's responding to the oxidative stress. And there's a couple ways that it's doing that. We have oxidative stress being produced as a result of xenobiotic metabolism. And everybody knows that and everybody thinks that. And when you think about dioxin causing oxidative stress, that's the first thing that triggers in your mind. Oh yeah, it's because of the P450 metabolism. But the other thing that the TCD receptor does is it induces this one gene down here called xanthine dehydrogenase. I talked about that before. It's responsible for taking xanthine and metabolizing that to urate. And urate's going to be dumped into the bloodstream. And urate, as I was talking to Dean about yesterday, is actually an antioxidant. So it's a secondary response in terms of the oxidative stress. Now, the problem is is in order to go to urate from xanthine, you have to go to hypoxanthine. And xanthine dehydrogenase actually has a second activity as well. It's the same gene, but it's two different enzymes. So xanthine dehydrogenase will use NADPH to go to urate from xanthine to urate. But for hypoxanthine to go to urate, you can either do it by xanthine dehydrogenase, or in under oxidative conditions, what will happen is that the xanthine dehydrogenase will be oxidated to xanthine oxidase. It no longer becomes NADPH dependent, but it's an oxidase now, and it's a huge producer of superoxide and hydroxide radicals. And so there, in itself, it's sort of trying to compensate for the oxidative stress, but what it's really doing is increasing oxidative stress as well. The second thing that's occurring is that you have an increase in glutathione production. And that increase in glutathione production, again, is in response to the oxidative stress that's being produced. So the NADPH pool is being required for the glutathione to support the glutathione peroxidase as well as the reductase. And so that's consuming that. And in addition, what's happening, in order to generate that glutathione, you're messing up one-carbon metabolism. So you have an increase in choline that's being produced as well. And that choline is actually being used to support the functioning of that. And that's what this gene is, that CTH that I couldn't remember, but I remembered it was involved in glutathione synthase or synthesis. So really, when you're talking about the pentose phosphate pathway and its increase as a result of oxidative stress, what it's really doing is it's supporting the production of glutathione, and that's how it's being responsive, as far as I can tell from the biochemical point of view. The second pathway I want to talk about is the disruption of carbohydrate and fatty acid metabolism. Let me know how I'm doing on time. And this one involves the TCA cycle, fatty acid synthesis, beta oxidation, and glycolysis, as well as gluconeogenesis. And so what we see is that there's a dramatic decrease in the acyl-CoA pool, and more specifically, acetyl-CoA. So acetyl-CoA, and that's consistent with the fact that there's no fatty acid synthesis that's occurring. You can see that there's these green boxes here. So the major enzymes involved in fatty acid synthesis are shut down. And the reason that they're shut down is because there's no acetyl-CoA to support that production. In addition, you have the green box here, and that's the first step and the rate-limiting step within beta oxidation. So beta oxidation is going to be shut down. Again, no source of acetyl-CoA. As a result of no source of acetyl-CoA, cholesterol metabolism is going to be shut down as well. But that's not the interesting thing. So the interesting thing that really stuck out and what drew us to this mechanism was this increase in oxaloacetate. And so I'm going, what the hell is going on with that, right? So the increase in oxaloacetate is this red dot right here. And if you look at that, that doesn't necessarily make sense because the TCA cycle is all ramped up. It's ready to go. It should be generating NADPH to support oxidative phosphorylation, right? But we know that there's oxidative stress as well. And for some reason, the mitochondria is just a target. It's just a sponge for oxidative stress. So you start compromising that situation, and you're limiting energy production, which is going to ramp up the TCA cycle. But in order for the TCA cycle to function, you need acetyl-CoA, because the acetyl-CoA is going to condense with oxaloacetate to go into the TCA cycle to generate NADH, right? I've got some heads nodding, and other ones are just sort of there. I got that deer in the headlight look, like it's 401, biochemistry 401 on a Friday morning at 9 o'clock. Okay. So you got this buildup of oxaloacetate, but oxaloacetate is also a precursor for gluconeogenesis. But the problem is, and this was actually done by Carl Rosman back in the probably early 80s, late 90s, that PEP-CK, which is the first step, just the first or the second step, in terms of taking oxaloacetate and pyruvate back to phosphoenol pyruvate, right? But it's down-regulated, so oxaloacetate can't go there as well. So this is why we see approximately a 40 or 50-fold increase in oxaloacetate. It just sort of jumps out right at you and hits you in the face. Now, the question then goes, well, okay, that's still fine. I mean, it's not like we're shutting it down totally, so there's some things that are getting through. But then you still have glycolysis that's occurring, right? And you've got a source of glycogen. And in the mouse, TCD actually causes a depletion of glycogen, so that there's some energy there and the animal should be somewhat happy, but that's not the case either. Because what's happening as, so you're not getting any reproduction or gluconeogenesis occurring here. So notice the glucose going that way. You're depleting the lactate as well, so the Cori cycle is all shut down because everything's getting combined or blocked up at oxaloacetate. As glycogen's being depleted, and as you're releasing glucose 1-phosphate and generating glucose 6-phosphate, it can proceed into the glycolytic pathway if it wanted to. But what it does is that gets then shunted off, remember, because of the pentose phosphate pathway. So that's what's occurring. What we have is we have a crisis in energy management, and the cell is not responding well to that, and as a result of that, we're going to eventually end up with some necrosis. As a result of that necrosis, we have immune cell infiltration associated with that, because it's going to clean up the mess. The cells can't survive, they're going to die, and we have this mess. And so what we did was we went to the gene ontology website, and we identified every gene that was associated with immune cell infiltration, and that was about 62 different genes. 84% of those contained a DRE, but only 18% had any kind of AH receptor associated with them. And the reason with that is because we're not really looking at a change in gene expression with this response. The change that we're really observing is that we have an infiltration of cells into the tissue that we're analyzing, so the increase in expression of these genes is a result of more immune cells, more macrophage coming into the tissue itself. And we haven't looked at the integrins and some of the other things that are associated with what you're talking about, but actually, that's the next slide. So the last slide is really the fibrosis. And so as a result of that immune cell infiltration, you're going to have activation of the stellate cells, and we've demonstrated that there's some fibrosis, and other people have demonstrated there's some fibrotic deposition. And so if you look at various genes associated with fibrosis deposition or collagen deposition, you can see that there's a dramatic increase. And sometimes what you'll also see is that there's a gene that's repressed. And in many cases, those genes are actually anti-fibrotic. So it's sort of a response to the deposition of the collagen as well. So it's consistent, but we can't say that. I can't say with 38 genes or in the previous slide for the 62 genes that we can explain all genes. I would say about 90% of them that make sense, and then there's 10% that seem like they're out of place. Okay, so we've gone through this, and we've talked about that. And so we've come up and we've proposed this mechanism of action in terms of the hepatotoxicity of TCD. What you really have is you have a situation in terms of oxidative stress. And that oxidative stress can come from a variety of different sources that I just talked about. So you have the accumulation of fats, we have the steatosis, and so lipid peroxidation, and even the metabolism, because you have a lot of changes of saturated fatty acids to unsaturated fatty acids, and that's going to create oxidative stress as well. You have an increase in iron into the liver as well. We never showed that. Actually, that's in the literature, but obviously you're going to have Fenton reactions, and that's going to create oxidative stress. We know from complementary studies that we have dose-dependent increases in TNF-alpha and IL-6, again, inducers of oxidative stress. And we have P450 metabolism. So it goes back to Niebert's studies where he did the knockout, and he showed that if you knock out 1A1 or you knock out 1A2, you see a diminution of the response, but not a complete abolition of the response. And that's because you have multiple sources of oxidative stress that cannot just be explained by P450 induction. So this oxidative stress then is going to cause a problem for energy homeostasis, and you eventually get depletion. And the cell can't survive without sufficient energy, and so it's going to die, and that's where you're going to get your tissue damage. And as a result of that tissue damage, you're going to have inflammation associated with that, and with that inflammation, you generate your fibrosis. And then what Gavin really didn't talk about yesterday was how fibrosis then leads to hepatocellular carcinoma. So, but that's, I guess, another talk. Now, as a result of this, and this is a limited study, again, this is actually based on microarray data, not the RNA-seq, we're working on that. We did dose response on all those genes that we could model, and we came up with EC50s for those genes. And what we found as well is that unlike other responses, and specifically like the estrogen receptor, we saw that there was this bi or trimodal distribution of EC50s for dioxin. Whereas with the estrogen in the uterus and even in the liver, everything followed one single dose response curve, and most genes fell along that. And if you look at this distribution, you see at very low doses, the first thing that you see is lipid metabolism. In the second group of EC50s, you have more of an immune response process that's occurring. And in the third set of EC50s, you see that you get the collagen deposition. So you have a stepwise progression of these events depending upon the dose. And what this tells me, and this is where you know, I might get into some arguments, is that there's a threshold with dioxin toxicity. And I'll even go, and I'll go really out on a limb because, you know, we're all friends, right? And that you will not get carcinogenicity at doses below this because there's not sufficient damage to create the responses required in order for the progression to get to that cancer. Now, that is under the assumption that the cancer that we're seeing is a result of the generation of fibrosis. So in summary, TCD disrupts at least 15 different pathways, and I guarantee you that there's more. And we're just sort of trying to build that out. And actually a really great way of doing that is Cytoscape, because that's where all those diagrams, and it helps you identify where to look and what things you may be missing in terms of your analysis. The progression of TCD elicited key events, and I chose that word key events specifically, is dose dependent. And so that just because you have steatosis doesn't necessarily mean it's going to lead to fibrosis and maybe hepatocellular carcinoma, because you have to trigger these other events to occur as well, and they only occur at higher doses. Now, we've done a 30-day and we've done a 90-day. Do we need to do a 2-year study to see whether even low doses will actually trigger each one of those events? And then the third thing is, this is the mouse, and the mouse is not the rat, and the mouse and the rat are not humans. And one of the things that I've also been looking at is the species-specific differences in terms of gene expression and DRE distribution. And so, although this is very interesting, I don't know the relevance of this in humans. Obviously, I didn't do this work by myself, and there was a lot of collaborators that were involved in this. And so here is a list. Jack Harkema was great in terms of the histopathology, and he's been very supportive of that. There's been multiple people involved in terms of the metabolomics, Sophia Lunt and Dan Jones, Nick Rio. The DRE work was done by Jason Matthews, who's at the University of Toronto. Gary Johns and Chris Jennings helped out in terms of the analysis of this and the Bayes analysis. Did I miss anybody? Dirk Colbury has helped out in terms of the high throughput analysis of the images, so that of the histopathology images, so we could actually quantitate the lesions and correlate those changes to the gene expression changes. And then lastly, the folks in the lab. So, and the primary folks that I'm going to talk about today are, work that I talked about today was Rance. So he's the Cytoscape guru in the lab, and has been great in terms of doing a lot of the computational work. Kelly's really helped out in terms of the in-life studies and getting those all done, because those are quite a bit of work. And then Agnes, who's now down at the EPA, has helped out. And then there was Ed Deere, who did the DRE search work. A lot of the lipidomics was done by Michelle, who's down at the EPA as well. Sun Tate actually did a lot of database work, and I'll wrap up there because I don't know what my time is like. But thanks, and I'll answer any questions if I can. Gavin?
I like a study, and I leads to two questions, and they're kind of related. Number one, when you ramp up xanthine oxidase to that level, you're going to also have metabolic demand by depleting the xanthine pools and by extension the ATP pools. So what happens if, have you tried giving allopurinol to the dioxin animals and see if that doesn't help? Yeah,
I mean, there's definitely different things that we can contribute and see whether it sort of alleviate some of the responses. But I think the problem is that you got 15 different pathways, and so that's just one component of it. And so it might be like a P450, 1A1, 1A2 story. You might sort of decrease it 10 or 20%, maybe 30%, but the other pathways may still be sufficiently functional that that's going to contribute and cause the response still.
What I'm wondering is the gene expression changes may do nothing if you don't put the metabolic demand on the organ. And that's why I'm asking. The other question is, a lot of times when you have that kind of metabolic stress, the cells will enter the apoptotic pathway, run out of ATP, and then undergo secondary, what's called apoptosis. Have you guys looked to see if there was any oncotic cell death, so that these very big hepatocytes that look like ghosts?
Yeah.
Very good signal for
Jack's looked for those and hasn't really seen any evidence, and we don't really see any evidence of any kind of apoptotic, apoptosis type of activity. Okay. Yeah. And so that's why we think it's more of a necrotic response.
Right.
Bob?
I have a couple of questions also, quick ones. This might be my ignorance, but you seem to be equating steatosis with lipid peroxidation. I'm not sure, is that really true? When you have steatosis, do you really get lipid peroxidation? And then my second question is regarding your EC50 slide, which I think was really interesting. Have you thought about those EC50s perhaps being related to the KMs of the rate-limiting enzymes in those pathways?
No, okay, I'll do the second question first. Yeah, because some of those some of the EC50s for those genes are obviously not going to be associated with, one, the KM, and two, it may not be a rate-limiting step as well. And I think it's more important in terms of a pathway to identify the KM for the rate-limiting step and what the EC50 is for that specific gene. And obviously, some of these responses are going to be not transcriptional as well, so that you're going to have changes in phosphorylation as a result of that. The second, or the first question, what was your first question again?
Whether steatosis is
yeah. Yeah, I mean, more recent evidence that we have, in terms of this more global type of non-targeted analysis by LCMS, MS, indicates that there's a significant increase in hydroxylated fatty acids, which would suggest the peroxidation is occurring.
But is that a consequence of the lipid accumulation?
I think it is. I think it's just mass action. The more lipids, the more targets, the more possibility that it's going to capture radicals.
Okay. Time for one more question.
So can you comment on that ascorbic acid that has been pumping the urine? Because it sounds to me counterintuitive that if you have a state of oxidative stress, that these animals are pumping out ascorbic acid into the urine.
Keen eye. Because very good eye, because I didn't talk about that. So what's happening with the ascorbic acid is essentially, and this is why I'm talking about the relevance of the mouse response to the human. Because what we see is that the mice have the capability of synthesizing vitamin C. Humans do not, okay? And what we see is that there's a dramatic increase in that synthesis pathway, and I never really talked about it, but that's why there's that increase. Now why the cell is responding that way, I don't know, but obviously it's dioxin, so you can't say that there's a physiological effect. But the vitamin C is also going to be important, the ascorbic acid is also going to be important in terms of the fibrogen deposition. So, it serves as a source, I think, for hydroxyl. It hydroxylates a proline within collagen that allows it to network and stuff like that. And it's also pulling away carbon from oxidative metabolism, a further sort of depletion of energy. So.
Thanks, Ken.
Sorry.
I might add that we did a metabolomic